you very much. Can you just let me know if you are seeing those slides?
Yes, we are. If you put them on full screen, perfect.
So yeah, thank you very much for the introduction, very kind, and also just for having the idea for the symposium, which was a great idea, and thank you for letting me be a part of it. So this is about PET, and we rather cheekily coined the term PET Metallomics. PET is positron emission tomography, and it's a slight change of direction. So we can answer some slightly different questions about trace metal biology using PET. So what you've heard so far, with all the amazing techniques that have been described, is information about what metals are bound to in cells, the role of metals in metalloenzymes, storage proteins, where they are in cells, where they are in the body, and how much, the different levels in different organs, and distribution among different cell types and within cells. And we've seen some amazing images that show that at the cellular level. And that levels of metals can be abnormal in disease and in dietary abnormalities. These methods represent snapshots in time. They are a picture of the result of prolonged accumulation and equilibrium of the metal with the tissue. One other thing that we know over the development of medicine is that chelation therapy and dietary therapy can be beneficial. But there's plenty of other things that we don't know. In many cases, we know that there's an association between abnormal metal levels and disease, but we don't know whether it's a cause or a consequence. We don't know an awful lot. We haven't methods to watch metals moving from one site to another, for example, from the diet to the tissue where they end up, or how they are excreted from those tissues. We would really like to be able to measure these things non-invasively in animals, or ideally in people, and we'd like to do it dynamically over time as opposed to taking a snapshot. And we'd also like to know if measurements of this kind could be clinically or diagnostically useful. So you've heard so much about the methods that operate at the cellular level for detecting, quantifying, localizing metals, and I don't need to go into that already, but I just want to emphasize that all of them rely on taking samples at autopsy or biopsy, and all of them give a snapshot as the end result. What I want to talk about is being able to measure the trafficking of metals dynamically and non-invasively in animals and in humans. And the technique that we've adopted for doing this is positron emission tomography. This is where we can use a radioactive isotope which emits positrons. It decays by positron emission. And the positron that's emitted is a little bit of antimatter, and very quickly it encounters an electron and annihilates with the electron matter to turn all of that matter into energy in the form of two gamma rays, which are emitted at 180 degrees to each other and are detected coincidentally by an array of gamma detectors around the patient. We can use a range of radionuclides, they quite routinely using fluorine-18, carbon-11, gallium-68, more recently zirconium-89, copper-64. All of these decay by positron emission, and they provide spatial information about where the molecules containing them go after they've been administered, and that spatial information comes from the collinearity of the two annihilation photons. PET has been a routine clinical tool now for between 20 and 30 years. And in summary, it tells us where the radioactive atoms are in the body, both spatially in 3 dimensions with a resolution of a few millimeters, and temporally and dynamically, so we can view the process of trafficking rather than just the end result. We can do the same with gamma emitters. It doesn't have to be a positron emitter. The only difference really is that there's only a single photon emitted. It's emitted by the atom itself rather than an annihilation a few millimeters from the atom. And the spatial information comes from collimation rather than coincidence detection. But again, the point is that we can find out where the radioactive atoms are in the body spatially in 3 dimensions, again with a resolution of a few millimeters. Here's a couple of examples of the kinds of images that you get. The first one on the left, and if I just give myself a laser pointer, the one on the left is an old one from the early 1990s of gallium-67 citrate, and this is a gamma camera image of a patient with lymphoma, and it's a planar projection, an anterior view showing the lymphoma here at the base of the neck, also seeing uptake of the radioisotope in the nasopharyngeal area, in the salivary glands, the liver and the intestines, and the mammary glands.
I don't know that.
So the one in the middle is more recently familiar. This is a FDG PET scan using glucose labeled with fluorine 18. This is the molecule here. And it gives us a picture of glucose metabolism. And this one is a coronal section, again, showing the three-dimensional nature. This is tomography. And the one on the right is an animal example, one of the perhaps the very first example of a scan on our preclinical PET scanners in our department at St. Thomas's, showing the distribution of a fluorine 18 labeled substrate of the sodium iodide symporter. So you're seeing uptake in the thyroid, salivary glands, stomach, and excretion into the bladder. So these are the kinds of images that you can get. As I said, we use a range of radionuclides routinely in nuclear medicine. Quite a few of them are metals, but the conventional way to use metals in nuclear medicine is by incorporating them into bioconjugates, such as this one here, which is a somatostatin targeting peptide with a chelator attached to it. And the chelator binds the metallic radionuclide, and the radionuclide is actually just a passive tag. It just follows the peptide around. It's the peptide that determines the biodistribution. So the gallium here serves as just as a radiolabel. What we're doing now in recent years in our group is turning the radioactive metals into a different use to monitor the biology of the metal itself, rather than just using it as a tag. So we're using it to image metal trafficking, and this is where I hope the technique would be of interest to many of the researchers we've heard from today, and many of the people in the audience as well. And we've called this PET Metallomics, just because it's nice to give something a name. It's becoming possible because we have an expanding array of positron-emitting radioisotopes of the biologically essential metals. And we have isotopes of copper that we've been using quite routinely for a few years now. Manganese isotopes are just emerging, and zinc. Cobalt and iron isotopes have been around for a long time. We haven't done any work with those recently. We can also use surrogates of essential metals. If we don't have the ideal radioisotope properties of the element itself, we can use a surrogate like rubidium-82 or thallium-201 as surrogates for potassium. Gallium-68 or gallium-67, which in some ways, not always, is a surrogate for iron. Strontium-85 is a positron emitter that can be a surrogate for calcium. And we can also use PET or gamma camera imaging to monitor the metal distribution after treatment with metallodrugs such as cisplatin using platinum 195M, or gallium drugs using gallium-67. And again, I want to emphasize the point that we're not just using these as tags, but to study the trafficking of the essential metal itself by PET or SPECT, dynamically and acutely in the short term, in living animals and in humans, non-invasively, for research and for medical diagnosis. So some of these radioisotopes are being produced at King's in the cyclotron, or one of the two cyclotrons at St. Thomas's Hospital. Available now, we have copper 64 and copper 62 with half-lives of 13 hours and 9 minutes respectively. Zinc 62 with a half-life of 9 hours. I'll come to that in more detail shortly. Manganese 52 is on the way. We've been working with manganese 52 produced by colleagues in Denmark. That source has dried up and we're working on producing it ourselves. Gallium, rubidium, all these are available now, and we're working to produce a wider range of radioisotopes, including zinc 63, manganese 51, possibly in the future lead 203, and eager to see ideas for other ones that we could produce as well, if there is a need. Some of these are produced, as I say, at the King's cyclotron at St. Thomas's. Some of them are produced in conjunction with an external partner who have at the University of Birmingham, who have a bigger and more potent cyclotron. And we're developing these as tools to support clinicians and metallomics researchers. So we need the input of people in our audience today. And this presentation is meant to show what is possible using PET by means of some examples. And I'm going to start with copper, because the work we've been doing on copper goes back a good few years now. I won't dwell on this. This is just to show the importance of copper, and you know this already from the earlier presentations. But copper is really important biologically as a constituent of a number of important metalloenzymes. And here's a bit of recent work showing what PET can do in humans. This is a study from a group in Aarhus who have done this imaging study in humans as a prelude to a clinical study in Wilson's disease. And they've compared the trafficking of copper 64 administered to patients by two different routes, intravenously on the top left or oral on the bottom left. And if you see what happens to the copper very quickly, within minutes, it is taken up in the liver. Nearly half of the injected radioactivity goes to the liver very quickly, and it's steadily excreted via the bile duct into the gut. And you can see that the excretion increases over time. By 20 hours, you can noticeably see that the copper in the liver has diminished. There's more in the gut, and there is recirculation of copper to the rest of the body, as has been mentioned by previous speakers, largely in the form of ceruloplasmin, as that copper is metabolized in the liver. If it's administered orally, rather less of it gets to the liver because a lot of it is not absorbed in the gut. It goes on simply to be excreted. And that radioactivity that is accumulated in the liver, some of that is also excreted back into the intestines for diminishing the amount of radioactivity in the liver, as well as recirculating to the rest of the body. The graphs on the right illustrate this in a quantitative form. So you see that intravenously administered copper very quickly accumulates in liver in a few minutes, and then begins to decay away as it's recirculated to the rest of the body. Rather less is taken up from oral administration. And the bottom graph shows the blood radioactivity, and clearly the blood is diminishing in radioactivity after intravenous administration as it's very quickly absorbed by the liver. But then the blood level rises again, as you can see, the recycling of copper back into the circulation in the form of ceruloplasmin. That promised Wilson's disease study hasn't been completed or published yet, but there's some work being done in animals and in a mouse model of Wilson's disease. Now, copper in the liver is normally transported to ceruloplasmin by a chaperone ATP7B. And these researchers at Texas used an ATP7B double knockout compared with the wild type, and you can see from the images that they're very similar to the human images. In fact, you get initial uptake after intravenous administration in the liver. The liver activity diminishes over a period of hours, but in the knockout mice, that liver activity does not diminish. And the quantitative graph on the right shows you the time course of uptake of activity in the liver in the wild type, where it very quickly is taken up and then stabilizes, plateaus, where accumulation and export are balanced, but the export is greatly reduced in the knockout mice, and it just continues to accumulate. So there's a very clear distinction between the two different genotypes. So the next steps, both for us and for our colleagues in Aarhus, is to clinically evaluate the copper-64 in Wilson's disease patients. We can also consider using copper-64 PET to monitor the effect of chelation therapy in Wilson's disease using chelating agents like tetrathiomolybdate, which is a recently developed one that's in clinical trials now. And the older chelation therapy agent that's been in use since the 1960s, penicillamine. And maybe we can also do that clinically and monitor the evaluation of monitor these chelating agents in humans as well. Turning to, staying with copper, but turning to cancer, and this is an example of a mouse scan from one of my PhD students, Joanna, who gave a mouse subcutaneous DU145 human prostate cancer tumor as a xenograft and administered copper-64. This is part of a bigger study, which some of which you'll see later on. But you can see uptake in that tumor on the shoulder. Again, you see the uptake in the liver and excretion into the gut. So there is a specific uptake of copper in the tumor. And this has been put to use in human prostate cancer imaging as well by this group in Italy. So just injecting copper-64 chloride intravenously into patients with prostate cancer shows that copper-64 is actually a rather good prostate cancer imaging agent. These are axial sections across the lower pelvis, and you can see uptake in the tumors. This is helped by the fact that there is very little uptake of copper-64 in this region in the background and in the normal prostate. So there's no radioactivity in the bladder to interfere with the image, and it's a high resolution image with good sensitive detection of prostate cancer metastases. Staying with cancer again, and we've begun to use this idea of PET imaging to explore sensitivity to drugs like cisplatin, drawing the link between copper-64 and cisplatin through the transporter CTR1, which is the main transporter of copper from the outside to the inside of cells. CTR1 has been implicated in many publications over the years as a significant route into cells also for cisplatin, the anti-cancer drug, suggesting that one possible mechanism for cisplatin resistance is downregulation of CTR1. So we hypothesized that maybe copper-64 PET imaging could predict cisplatin resistance. Now, cisplatin is an important mode of therapy in ovarian cancer, and a large fraction of ovarian cancer patients become resistant, maybe 70%. So the question was, could ovarian cancer might be a good model for this, and with a clinical implication, if it works, that we could stratify patients into those who will respond to cisplatin and those who won't, which is rather nice if you can avoid treating with patients with cisplatin, which has very unpleasant side effects because it won't work. That would be a valuable outcome. So we had a look at the ovarian cancer model cell line A2780, which has a platinum-resistant counterpart, A2780 cis, where the resistance is induced by repeated passaging in the presence of cisplatin. And just to check out that there is a change, a downregulation of CTR1 in these resistant cells, we measured CTR1 resistance in relation to the quantity of cells, both at the protein level and at the transcript level, and showed that there is knockdown of CTR1. And the knockdown of CTR1 influences uptake of copper-64 into the cells. It reduces uptake significantly. And in the normal non-resistant cells, the CTR1 can be blocked by reducing its expression using siRNAs. The copper-64 uptake in vitro then goes down. There is less of an effect in the resistant cells because they're not expressing so much CTR1, and the copper that does get into the cell, which is rather less, is getting in through other routes. So certainly there may be other contributions of other mechanisms for platinum resistance, but certainly CTR1 downregulation is one of them. The data on the right shows what happens to the cisplatin accumulation in cells measured by some of the techniques that you've heard about already today. Cisplatin accumulation in the cells is significantly suppressed by about 70 or 80%. That is due to the suppression of the uptake, not due to any change in the retention, because the retention in both cell lines is pretty similar. At the same time, we investigated copper-64 uptake, and that same pattern is seen. The uptake of copper-64 is severely diminished by about 80% in the resistant cell line. And again, that is not related to excretion because the efflux from copper from the cells is the same in both cell lines. So all is very hunky-dory in vitro. There is a clear parallel between copper uptake and cisplatin uptake. What happens in vivo? Well, rather disappointingly, first of all, the image is shown on the right with the A2780 tumor on the left and on the flank here, and the A2780 cis. Both are visible in the scan. You can't really visually see much difference between the two scans, but collecting quantitative data over a number of mice, you see, actually, rather sadly for us, the situation is reversed in vivo. There's not much difference between the two, and actually the uptake is more in the resistant cell line than in the unmodified cell line. So it seems that the in vitro interpretation is a major oversimplification of what might happen in vivo. Also, we conducted similar in vitro experiments with patient cancer cells from patients who had or had not become resistant. And again, we didn't see any consistent trends in relationship between platinum and copper uptake in those cells. So it appears that this wasn't a great hypothesis. The prospects for human diagnostic use of this idea seem to be fairly limited. And it's really because the in vivo environment is a lot more complicated than in vitro. In many ways, one of the ways we investigated to see, just one of the many possible ways, was just to see what the effect of serum is. So those in vitro experiments on the previous slide were conducted in 10% fetal bovine serum. We repeated some of those experiments using 100% human or mouse serum as the medium, and found that in every cell line, whether resistant or not, the copper uptake was very, very much suppressed by the presence of 100% serum instead of 10% serum. So that's just one of the many possible interferences that there could be because of the complexity of the in vivo environment. Several more are listed in the box in the middle. So I want to switch away from copper to some work that we did a few years ago on Alzheimer's disease models. And many papers over the years have suggested that copper trafficking is altered in Alzheimer's disease. And I've put the word altered there and deliberately chosen a vague word and put it in inverted commas because the literature is very mixed up on this subject. Many papers say the total brain copper load increases in Alzheimer's disease, or the intracellular copper decreases in Alzheimer's disease. And for every paper on one of these, there's a paper saying the opposite. In vitro, it's known for sure, though, that amyloid beta and amyloid plaques can bind copper. And when they do, aggregation of the amyloid beta is promoted and free radicals are generated. We also have data to indicate both preclinically and clinically that treatment with lipophilic chelating agents like cleoquinol, PBT2, and GTSM, and confusingly their copper complexes, can improve cognitive function and decrease plaque burden. And a little bit of copper-64 PET work has been done in animal models of Alzheimer's. So Paul Donnelly and co-workers at Melbourne University looked at copper-64 GTSM in APP-PS1 mice and showed an increase in uptake of copper-64 compared to normals. So we hypothesized that PET with copper isotopes could be a useful imaging biomarker for Alzheimer's disease progression, possibly response to treatment. And maybe also copper-64 PET could be used to determine the effects of chelating therapy on copper distribution and on the delivery of copper via copper chelates. And this work has been reported in a couple of papers that you can see there. We chose to use a different model. This is the TAS-TPM model developed by David Howlett at King's. This is a cross between two mutations, which results in amyloid plaque deposition over a period of months, and neuroinflammation and cognitive impairment. And so we used this model partly because of local availability and partly because it was different to the previous one. And we began by looking at copper-64 chloride, and you may have noticed in the images that I showed earlier that there was no significant uptake visible on those scans in the brain, whether in the animals or in humans, after copper-64 chloride administration. But fortunately, the postdoc doing this work, Erica Andriozzi, was a little bit more persistent, and she took a closer look at the brain, and it turns out that it wasn't completely free of copper uptake. There was a tiny bit of copper uptake, but very highly localized to the areas around the ventricles. So what you're seeing in the image on the left here is a sagittal section through the mouse brain. So you can see in the CT scan the cranium here, and you can see hotspots of uptake which correlate quite close to the ventricles. And here's a coronal section showing the same thing. And this is confirmed. This is really pushing the limits of small animal PET here. But this we can confirm this with autoradiography of brain sections. So these hotspots here are actually digital autoradiographs, again showing that radioactivity is confined to areas in and around the ventricles. But the uptake is very low and not enough to study differences between Alzheimer's and normal. So copper-64 chloride PET is unlikely to be a sensitive biomarker for Alzheimer's disease-like pathology, simply because you cannot get enough copper into the brain to image it adequately. But we have an alternative. So for many years, we have been, 25 years or so, we've been working with copper bis thiosemicarbazone complexes labeled with copper-64. And these allow us to control the biodistribution by taking advantage of the redox activity of these complexes. They're all lipophilic. We've made dozens of these derivatives of these complexes. They can all penetrate tissues and get into cells in proportion to blood flow, independently of transporters, and they release copper upon entry into cells by a bioreductive mechanism. That is to say, some of them do. We can control that bioreductive mechanism to prevent this using copper ATSM, which gives us hypoxia selective uptake. And if you inject this into normal mice, you don't, although this tracer can cross the blood-brain barrier, you don't see any uptake in the major tissues, even the ones with high blood flow like heart and brain, because they're not hypoxic. We have another type of these ligands which are not hypoxic selective. They're much more easily reduced, and the copper is released immediately upon entry into any kind of cell, regardless of whether it's hypoxic. And you can see the difference in the biodistribution here, because all of the organs that have high blood flow have high uptake. The heart has got a huge uptake of this tracer, the lungs, even the brain has got quite a bit of uptake and trapping. So this is a useful way to deposit copper in the brain and get it released from the chelator as soon as it gets into the cells, and then we can monitor its efflux, and there's enough copper there to do quantitative studies. So this is a comparison of this with copper GTSM on the right, with copper acetate, familiar distribution in the liver and excretion into the gut for copper acetate. The copper GTSM goes to the heart and the lungs, also to the liver and excreted through into the intestines. By 24 hours, most of those tissues have lost most of their copper. So the heart has lost a good deal of its copper by 24 hours, but noticeably the brain hasn't lost any of its originally taken up radioactivity. It reaches a peak reaches a plateau at about 30 minutes after injection, and it stays there, which raises the interesting question, which I don't have an answer for, and maybe this, maybe it can promote a bit of discussion, but does the brain lack an efflux mechanism for copper? The data are summarized quantitatively on the left. The copper GTSM, you can see the heart seriously loses about 2/3 of its copper over the first 24 hours after that first 30 minutes. The lungs do as well, and the kidneys, but the brain and the spinal cord keep the copper that they have acquired in that first 30 minutes. The same is true in the brain of animals given copper acetate, but the uptake in the first place is so low that it's very difficult to quantify it. So this is copper GTSM comparing the Alzheimer's disease model versus normals. And just visually, you can look at these. These are close-ups of the brain, a coronal view and a sagittal view. And one thing that you notice in a qualitative sense is that there is widely distributed copper across the whole brain, but it's a little bit more heterogeneous in the Alzheimer's disease model. And this isn't just a visual interpretation. This has been confirmed by quantitative measures of texture analysis as well. So there's one noticeable feature of the Alzheimer's brain is that there's a greater heterogeneity of uptake and more patchiness. On the right, the bottom graph shows you that as in Paul Donnelly's results, we saw an increase of the initial uptake in that 30-minute period in the TS-TPM brain compared to the wild type, about a 40% increase, similar to what Donnelly observed. And the graph at the top shows the clearance from the brain between 30 minutes and 24 hours. And again, in the wild type brain, those lines are horizontal. There's no noticeable clearance, whereas in the TS-TPM brain, there is some clearance, not much, but it's about 5 times more than in the wild type brain. So there we have a metal fingerprint for this particular model of Alzheimer's disease. We can see increased heterogeneity, increased uptake, and increased clearance. 3 different parameters. Another thing we looked at, just to finish this section on Alzheimer's disease, was to see whether there was any correlation between the location of the radioactive copper and the amyloid plaques, which we measured by Congo red staining in tissue sections. It may be quite difficult to see from these images, but you can see the amyloid plaques stained in little red dots in the top image. And we found no obvious positive correlation between the distribution of copper and the distribution of amyloid, and maybe even a marginal negative correlation. So again, a bit puzzled by that. We thought we might be clarifying the situation with regard to copper's relationship with Alzheimer's disease. So far, we haven't really clarified very much, but we have found a fingerprint. So moving on to zinc. Zinc 63 is the radionuclide that has been used recently by other groups. It has a very short half-life of 38 minutes, so it's quite limited in what it can do. But DeGrado and colleagues at the Mayo Clinic used it to study zinc trafficking in animals and like us, in the brains of Alzheimer's patients. We have developed a production of zinc 62 in collaboration with the University of Birmingham, designed really as a precursor to copper 62, which was our interest. But since we had made the zinc 62, and it has a longer half-life of over 9 hours, which gives us a lot more time to do interesting in vivo studies, we decided to have a look at it for imaging. And nobody has done this before, even though zinc 62 has been available, because of fears that the imaging ability is compromised by the fact that it decays to copper 62, which is also a positron emitter with a much shorter half-life of 9 minutes. And nearly all of the positrons that you would get from the scan would come from the copper. But since we were already making it, we decided to take a second look at this idea. And we reasoned that because of the ratio of the half-lives, 9 hours for zinc, 9 minutes for copper, is a ratio of 60:1. And if you do the maths, after the first hour post-injection, the remaining radionuclide in vivo has spent an average of 98% of its time as zinc before it turns to copper and the positron is emitted. So it should really reflect the biology of the zinc rather than the copper. We set out to see whether this was a realistic view, and if so, zinc 62 would be a useful radionuclide to monitor zinc biology. So this is George Firth's project, who's a current PhD student in my group, and he compared zinc 62 citrate with copper 64 citrate to see if there was a substantial difference between the zinc and the copper. And first of all, the things that you notice that both of them have a high liver and abdominal uptake, as you see uptake in the liver and excretion into the gut. The one on the left has got very poor resolution compared to the copper 64 because the positrons emitted by the copper 62 have a much higher energy and they travel further before they are annihilated. There's also a hint in the images on the left of pancreatic uptake. And this is confirmed in the preclinical studies from DeGrado and co-workers, who also saw a high activity in the pancreas, even higher than in the liver. And that is not shown in the copper images. So we fully quantified these both from imaging and from dissecting the animals post-mortem and counting the tissues. And again, these results show very clearly that there's very high uptake in the pancreas of the zinc 62, but not the copper 64. So the zinc 62, copper 62 scan reflects the biology of zinc, not copper. And it has the advantage of this 9-hour half-life because there's much higher activity in the pancreas, and you can see it also in the spleen and the bone and the brain and the salivary glands. It's different in all of them. So the conclusion from that is zinc 62 is a valid tool for imaging zinc trafficking in vivo. And that's what we will be doing over the next few months to take this into application in cancer applications and possibly in applications in diabetes and so on. Here's an alternative method for measuring metals in tissues, instead of using the radioactive metal itself, in principle, it might be possible to use a radio-labeled chelating agent for the metal. And this is again work by George Firth and Graham Stasiuk, colleagues in our department, although this work was done before they came to King's at University of Hull. They've got a chelator which binds selectively to zinc and is fluorescent, and its fluorescence changes when it binds zinc, and also its solubility changes when it binds zinc. The ligand itself is lipophilic and can penetrate cells. When it binds zinc, you can imagine it binding zinc inside a cell and becoming cationic and therefore unable to easily escape from the cell. So the ligand would then accumulate in cells in which there is a high concentration of zinc. And you can see that this works in the fluorescent images at the cell level below. You can see that under zinc-free conditions, some probe, but not so much, gets into the cell. And some of it binds to zinc. If you pre-incubate the cells with zinc to load them up with zinc, you get more probe going into the cell, more trapping of that probe, and the trapping is because it is bound to zinc, and you can measure the zinc-bound probe separately. And this ligand has been labeled with fluorine 18, and we've done the first biodistribution studies with this. We don't yet know if this biodistribution on the right really does reflect in any way the biodistribution of zinc, but I'm showing you this is very preliminary as an alternative way to monitor the distribution of trace metals using PET. Emboldened by the conclusion that zinc 62 is a good marker for zinc distribution, we looked at zinc 62 uptake in vitro in a range of cancer cell lines. Again, this is work by George Firth. And in all of the cell lines, 5 of them that he looked at, and a 6th was a prostate epithelial cell line, which has extremely high zinc uptake. All of them took up zinc very extensively and very rapidly, maybe easily 30 to 40%, even higher after a longer period of the available radioactive zinc. And the uptake of zinc was maybe 10-fold higher than the uptake of copper. So there's a good possibility that we'll get enough zinc into tumors to be able to measure the changes in zinc as a result of different phenotypes and different metabolic conditions. So we took this in vivo, and just to see what it was like in a breast cancer cell line, MDA MB231, again implanted on the shoulder flank. Fairly soon, hours to a day after injection, for both zinc and copper, we see uptake in the tumor, as well as the usual uptake in liver and intestines. The images are improved at 24 hours because the background has subsided. But there is clearly the capability to image the tumors, which would lend itself in the future to quantifying the response of uptake of zinc in the tumors to different conditions. A little bit of work has been done on manganese. A very little bit of work has been done on manganese, in fact, and that probably reflects the amount of work that is generally done by other metallomic techniques on manganese. Manganese is far less explored so far than copper and zinc, and that is true also in the PET world. So George and Julia replicated some work in the literature by looking at manganese 52 chloride injected intravenously, observing, again, we see this familiar pattern by now, very high uptake in liver and excretion into the intestines, but also in the short term, for the first few hours, uptake in the myocardium, growing uptake in the salivary glands. By 24 hours, the myocardium is cleared, but the salivary glands haven't. Another thing that if you look hard, you can see that there is a noticeable uptake, which may well correspond to the pancreas. And that persists at 2 days, maybe even 3 days. So there are interesting features of the distribution of manganese changing over time. And again, if we quantify this, you can see that there is clearly, this is quantified by dissecting the tissues post-mortem, very high uptake in the pancreas, higher than in the liver, and also in the salivary glands. So there is a prospect of using manganese for looking at the physiology of the pancreas, for example. Rubidium is another one of interest because it's well known actually as an imaging agent in nuclear medicine in cardiology for imaging perfusion of the myocardium. So we get these kinds of images here. So this is an axial section of the myocardium. You can see the left ventricle here, and you can actually see a little bit of uptake in the right ventricle as well, and also coronal section, a sagittal section here. But this works because rubidium is taken up into myocardial muscle cells by virtue of being a substrate for the sodium-potassium ATPase. It's a potassium analog, which is very highly abundant in the heart. So this uptake reflects metabolism as well as blood flow, and it should be possible to dissect out the component that is due to the sodium-potassium ATPase expression. And we reason that this would also be interesting in cancer because sodium-potassium ATPase does vary in cancer and can be a marker for sensitivity to different drug treatments. And so in collaboration with colleagues at University College London, Ashley Groves looked at, while studying patients in both cardiology and cancer, looked at some patients with, well, a range of cancers actually, but this one is a colorectal cancer. So you see at the bottom here, cross section in the lower pelvic area, and you see high uptake of the rubidium-82 in the tumor. Some of that uptake reflects just increased perfusion compared to the surrounding tissues, but some of it doesn't. It probably reflects changes in the activity of the sodium-potassium ATPase. So there is a prospect for being able to measure the function of this transporter in different disease contexts. How do we know it's a substrate for the sodium-potassium ATPase? Well, colleagues Rick Southworth and Tom Eakin have performed isolated perfused rat heart experiments to measure the radioactivity of the myocardium after rubidium-82 is allowed to flow through the arteries. And in the control unmodified heart, you see there is initial rubidium-82 flowing through, and then it stabilizes with some trapping of radioactivity in the heart. That trapping can be increased by stimulating the sodium-potassium ATPase with isoprenaline, or inhibited, reduced by inhibiting the sodium-potassium ATPase with ouabain. So there is an element of measuring these transporter activities with rubidium. Finally, a look at gallium. Gallium-67 and gallium-68 are both useful radionuclides in nuclear medicine. The old application of gallium-67, which has been around for 40 years now, is in imaging lymphoma. And this works because it binds quickly to when gallium citrate or any gallium salt is injected into blood, it quickly binds to transferrin, to the ion binding site of transferrin, and becomes delivered to transferrin receptors. And in tissues like lymphoma, where there is increased transferrin receptor expression, we see uptake, and this is diagnostic of lymphoma. Another application which is emerging, this is not an old application, it's a new one, is imaging infection, imaging the actual presence of bacteria and fungus. And that's because these organisms secrete, well, they need iron just like we do, and to get it in low iron environments, they secrete chelating agents called siderophores, which have a very, very high affinity for iron. And they can capture the iron, and the complex is then recognized by a receptor on the organism's surface and internalized. Well, gallium is very similar in its structural coordination chemistry to iron. And it forms more or less equally high affinity complexes with the siderophores, and they have the same structure as the iron complex. So they too are recognized by these receptors when they have been complexed by siderophores. And so we have the potential, if we can make complexes of this kind with gallium-68, to image the presence of bacteria themselves very specifically. Now, a number of these siderophores have been evaluated preclinically, and we are on the point of evaluating them clinically as well by taking advantage of the fact that desferrioxamine, which is an important siderophore, is an approved drug for treatment of different kinds of iron overload. So it has the regulatory barriers to getting this into a clinical trial are quite low. Preclinical work has been done by colleagues in Serbia. So this is an animal that has been infected with Pseudomonas in the flank here and Staphylococcus aureus in the flank here. And you can see positive images very soon after injection with gallium, the gallium-68 desferrioxamine complex. And our contribution here has been to make a GMP compatible version of the gallium-68 complex, which we can use in humans. And a clinical trial is about to start. In fact, it was about to start a year ago if we hadn't been delayed by COVID, but it's ready to go now, led by Sally Barrington and Marlene Fisher, to look at the uptake of this tracer in patients with infections. So just to summarize then, what I've wanted to show you, and I hope done at least made some progress in that endeavor, what Metallomics can do for you as an audience interested in metal biology. It can help you to translate basic science in vitro at the cell level of trace metal metabolism to the in vivo whole body level. And it can also help you to translate from static snapshots of accumulation of metals over a long period to dynamic acute processes. We can measure the effects of disease on trace metal metabolism. We can diagnose disease. There may be opportunities for new types of diagnostics that we can use imaging for. We can monitor mechanisms and effects of therapy, potentially. I haven't shown examples of that yet, but we have been working on this in chelation therapy. Determine pharmacokinetics of metallo drugs like gallium and platinum drugs. And again, I haven't showed you examples of this, but this is something that we have been working on. We can help in principle help understand trace metal dietetics and delivery of dietary trace metals, and also potentially another aspect of Metallomics that has also been mentioned today, the trafficking of environmental toxins. Again, there are ways we can apply PET to this. So we can't just do this all by injecting trace metals intravenously. We have to be imaginative to produce a range of new radiometals, administer radiometals in different chemical forms by different routes to answer the questions, and develop new chemistry to do this. We may need to monitor the influx and efflux of metals into tissues separately, and we can exploit subtle differences between metals and their surrogates to answer key questions as well. So in summary, we can develop in vivo imaging tools to support Metallomics community. You as that community can help us by giving us questions that we can help you to answer. And I'd like to thank a huge number of people who've contributed to the work that I've shown here and work that I haven't shown here in different aspects of the Metallomic PET studies, and quite a large number of funding agencies as well, including the Wellcome for funding the London Metallomics facility. I'd particularly like to thank Thea for being such a potent driving force for the London Metallomics facility. Since she joined, it really has taken on a life of its own and grown out of control. And this is a wonderful thing. And the effort that she's put in and the example she showed is fantastic. So thank you very much. And thanks to KCL for the generous support in backing up that London Metallomics facility grant. And thank you guys for your attention.